Identifying a plant involves more than observing whether an organism grows from the soil. For centuries, scientific classification relied on a simple but flawed assumption. If it didn't move, it belonged in the plant kingdom. Biology operates as an evolving map, requiring constant redrawing to match new discoveries. Massive sections of the botanical world were violently pruned away. Analysis of cellular machinery eventually forced a divorce between entirely different domains of life. Fungi, characterized by chitin-based walls, and cyanobacteria, once called blue-green algae, were formally evicted from the plant kingdom. This taxonomic correction is now standard documentation, following R.H. Whitaker's 1969 system. The distinction is cellular. If an organism lacks cellulose walls or specific chloroplasts, it is no longer defined as a plant. Early botanists relied on gross superficial morphology. They categorized the natural world based on what was visible to the eye, habit, color, and leaf shape, missing the deeper biological evidence. Modern classification requires looking past these surface traits. It is a process of uncovering the structural truths that define an organism's actual evolutionary lineage. Carl Linnaeus pioneered this surface-level approach through early artificial systems. These systems assumed all visible traits carried equal importance, giving the same weight to vegetative traits as to sexual characteristics, ignoring that vegetative traits are easily altered by environmental stress. Natural and phylogenetic systems eventually replaced this framework. Instead of grouping plants by appearance, scientists began tracing shared evolutionary ancestors. Researchers now operate at the molecular level, mapping family trees using massive datasets. We scan the chemical nodes and DNA sequences operating inside the cells to confirm ancestry. This practice is called chemotaxonomy. Analyzing specific chemical constituents, such as unique proteins or metabolites, allows scientists to resolve disputes that visual inspection cannot settle. Reading this internal code allows us to reconstruct the specific engineering journey required to move life from the ocean onto the land. Algae represent the foundational architecture for all plant life, aquatic, autotrophic, and structurally simple. Their global impact is massive. These organisms are responsible for fixing at least half of the total carbon dioxide on Earth through photosynthesis. Morphologically, algae span a wide range. Microscopic species like Volvox form simple, free-floating colonies. Larger marine forms like Fucus show structural differentiation, developing a holdfast to grip rocks, a stipe, and leaf-like fronds for light absorption. Despite these distinct parts, the organism remains a thallus, a body lacking true roots, stems, or leaves. The thallus is an efficient engineering solution for a buoyant environment. Surrounded by water, it has no need for rigid, woody stems to fight gravity or deep root systems to seek moisture. Algae diversified into 3 distinct classes based on internal upgrades to their pigments and food storage systems. These evolutionary paths are categorized by the biochemical data shown in this table. Green algae, or Chlorophyceae, utilize chlorophyll A and B, storing their energy as rigid starch, a system optimized for stable, shallow freshwater. Marine intertidal zones present different challenges. Here, organisms face constant wave action and the threat of drying out when the tide recedes. Brown algae evolved a specialized cell wall coating to handle this exposure. This coating is algin. It is a gelatinous hydrocolloid that serves as a moisture shield, preventing desiccation while the plant is exposed to the air. In the high-pressure, low-light environments of the deep ocean, red algae utilize Floridean starch. Its molecular structure mirrors animal glycogen rather than standard plant starch. Biological color, green, brown, or red, serves as a visible proxy for specialized chemical tools engineered for specific depth, light, and pressure constraints. Before plants could colonize the land, they had to solve a critical problem: protecting their reproductive material from the open air. While early algae released identically sized cells into the water, evolution eventually favored a highly asymmetrical reproductive strategy. This strategy is defined as oogamous reproduction. This approach dedicates maximum resources to a single, large, nutrient-rich female gamete, while minimizing the size of the mobile male gametes. The development of a stationary, protected egg was a necessary prerequisite for any plant lineage attempting to survive in a dry terrestrial environment. Bryophytes, the mosses and liverworts, represented the first successful evolutionary step out of the ocean and onto terrestrial rock. Because they exist between aquatic and terrestrial worlds, they are formally designated as the amphibians of the plant kingdom. Their ecological role is that of a pioneer species. They are capable of colonizing bare, hostile rock surfaces where soil has not yet formed. Survival on land required a physical upgrade. They replaced the floating algal thallus with a prostrate body that hugs the ground to conserve moisture. To counter the forces of gravity and erosion on bare rock, bryophytes evolved specialized structures for anchorage. These are rhizoids. They are simple, multicellular attachments that secure the plant to the substrate, despite lacking the water-transporting tissues found in true roots. Rhizoids provided the first vital anchor point, allowing biology to permanently grip terrestrial geography long before the evolution of tall stems. Despite their land-based lifestyle, bryophytes are limited by a reproductive system still engineered for the ocean. On a liverwort, specialized vertical structures rise above the ground-hugging body, specifically to shield reproductive cells from the air. The archegonium is a flask-shaped female sex organ designed to encase and protect a single egg from drying out. While the egg is protected, the male gametes, or antherzoids, still rely on flagella to move. They have no mechanism for travel other than swimming. Fertilization on land requires a temporary physical bridge, a thin film of water from rain or dew, to allow the gametes to reach their destination. This physical tether is absolute. Without a droplet of water connecting the organs, the antherzoids cannot complete their journey, and the reproductive cycle fails. The plant kingdom represents an ongoing, verifiable record of organisms fighting a brutal biomechanical war to escape the ocean, one cellular upgrade at a time.